Hello everyone, welcome back to Chinmoy's Biology channel to a new episode of sickle cell anemia. So in the previous episode we have already discussed about haemophilia in details, and it will be displayed in the description box the video link if you have not yet gone through that lesson. So today's lesson will be in details about sickle cell anemia and the first disease, that is autosomal recessive disease, you are going to study today. So please don't skip in the middle of the episode because it is going to be a very interesting as well as informative episode. So stay till the end. Let's get started. So let's start this lesson with few questions. Now you all know that our red blood cells are biconcave shape or round shape. So what if their shape suddenly changes to sickle shape? What will happen? And how these sickle shaped cells will affect our body? Will they cause any problem? Will they cause any disease or deformity in our body? Let's check that first, and then we will come to the disease called sickle cell anemia. So you can see here, these are the normal RBC which are biconcave in shape, and this is the sickle cells, so which have changed their shapes to sickle shape, right? So this is a patient of sickle cell anemia, this type of cell they are having, and this is a normal healthy person. So what deformities they are causing in their body, so how it is affecting our body, I'll discuss first that. Now, how does it affect the body? Let us discuss this one first. Then we will come to that what is the disease. Now, the problems caused by sickle shaped cells, what is having, it is clogging the blood vessels. That is, if these are the normal blood vessels, if suppose this biconcave shaped RBC is flowing through this blood vessel easily, when it is sickle shaped, it will get stuck in some parts of a narrower parts of the blood vessels, so it will get stuck first of all. So when it is getting stuck, the hemoglobin is not reaching, as it is RBC only, so RBC is carrying the oxygen, so the oxygen is not reaching to all parts of the body, so reduced oxygen supply will happen in our body. There will be pain because these cells, they get ruptured, oxygen doesn't reach to certain parts of the body, which causes pain, swelling, or we can say that we some kind of discomfort in those parts of the body where the RBC is not flowing properly. Okay, so it has got stuck in some part of the vessel, causing this type of discomfort. Now, breakdown of RBC, so at times these have a very less life, so they we can say that they get destroyed very easily, so that causes anemia, and in term it causes fatigue and paleness. So if a person is anemic, it causes fatigue as well as paleness. Now, it also causes certain organ damages. Spleen, liver, brain, kidneys are always affected with this disease, sickle cell anemia. So what are the symptoms? How can we identify that a person is having sickle cell anemia? Because fatigue will be there, joint pain, swelling in hands as well as feet will be there, delayed growth will be there, and frequent infections. A person with sickle cell anemia will have very frequent infections. So this is how this sickle cell anemia is affecting our body. So now we will come to the point that what is sickle cell anemia. So, what is sickle cell anemia? So, as the definition suggests, sickle cell anemia, it's a genetic disorder where the RBCs become rigid and sickle shaped instead of round and flexible. Now, what happens? Why all of a sudden the round RBCs, they are becoming sickle shaped instead of round? So, mutation is in the HPB gene on chromosome 11. It is happening. So mutation and all in details we will be studying in the next chapter. So mutation in HBB gene on chromosome 11 means some changes in the protein sequencing happens. So in the next lesson only we will study about the molecular biology. So we will study about the protein synthesis, replication, transcription and translation. But here let us get into the brief of this. So what is HBB gene? It is also known as the hemoglobin beta gene, and it is located on the chromosome 11 and codes for the beta globin protein. So it is coding for the beta globin protein, and beta globin protein, what it is doing, it is combining with the alpha globin to form hemoglobin. So beta globin protein is combining with the alpha globin to form hemoglobin, and which is a major component of the red blood cells, and it carries oxygen throughout the body, and it is actually carrying oxygen throughout the body. Now, we can say that this mutation or change in the HBB gene in the chromosome 11 is causing the producing the abnormal hemoglobin S instead of the normal hemoglobin A. Okay, so this is the sickle cell hemoglobin and this is the normal hemoglobin. So how it is formed generation after generation? It is a autosomal recessive disorder. That is, we will check in the next slide using the Punnett square. If both the parents are having recessive genes of hemoglobin sickle cell anemia, then we can say that it will be inherited in the next generation. So let us see in this Punnett square what is happening. So let us see in this Punnett square, so if we are seeing the parental cross, both the father and the mother, they are having this as a carrier. Okay, so this sickle cell, it is there present in the gene as a carrier, and it is we can say that both are carrier and it is a recessive trait. And after the cross in this Punnett square, suppose here is male and here is female. So the first offspring is normal, second one is a carrier, HA and HS, third one is also a carrier, and fourth one is the affected individual. So you can see the cross. So the genotype will be 1 HAHA, 2 HAHS, and 1 HSHS. So what is the phenotype? Phenotype is 1 normal, 2 carriers and 1 sickle cell sufferer. So this is how it is working, as it is an autosomal recessive thing. If it is present in both the parents only, it will be represented or expressed in the next generation. If one is a carrier, if other is not a carrier of this disease, it will not be represented as a dominant thing. It will just carry on as a carrier. Okay, so here it is present in both, and so after the Punnett square, it is having 1 normal, 2 carriers and 1 sickle cell sufferer. Okay, is it clear to all of you? Now, just as per the Punnett square, we already know that the disease is controlled by a single pair of allele. That is, this hemoglobin is normal and this hemoglobin is the sickle cell hemoglobin. So out of the 3 possible genotypes, only the homozygous individuals of HBS, they show the diseased phenotype. So if both are HBS and HBS, they will only show the diseased phenotype, whereas heterozygous HBA and HBS individuals, they will apparently be unaffected, but they are the carrier. So this is what we have spoken about in the previous slide, where we have done the Punnett square. So HBA and HBS, heterozygous individuals will be carrier, but they will not be the affected one. But both who are homozygous will be the diseased phenotype, right? Now, therefore, there is a 50% probability of transmission of the mutant gene of the progeny, thus exhibiting sickle cell trait. So, if both are, just now we have told that if both the parents are carriers, only 50% chances are there of the progeny exhibiting the sickle cell trait. Okay. So, now we will move on to the next other parts, where we will discuss about the symptoms and other things of sickle cell anemia. Now, what is the molecular basis or scientific explanation of sickle cell anemia? Now, the defect is actually caused by the substitution of glutamic acid by valine in the beta globin chain of hemoglobin. So, what is happening here? You can see here the glutamic acid was there and it is replaced by valine. Okay, in DNA, it was GAG and CTC. So here the DNA has, in the sickle cell, a mutation has happened in the DNA strip. It has become CGTG and CAC. So this mutation has caused a change in the RNA sequence. So RNA, in place of GAG, it has become GUG. So when it has become protein, or we can say that amino acid, so amino acids are only forming protein. So when it has become amino acid is glutamic acid instead of valine. So where it is happening, this thing is happening in the 6th position of the beta globin chain. So this is a beta globin chain and it has occurred in the 6th position. So this is the 6th position of the beta globin molecule or chain of the hemoglobin molecule. So this, like this, and when we are studying about molecular biology, in molecular biology we will study the formation of from DNA, RNA, mRNA, then protein synthesis and all we will study in detail. So that is called the central dogma of molecular biology. So that in the next lesson we'll study. So for that you need to subscribe to my channel and stay tuned for what all interesting topics I'll be teaching. Now let us come back to this. Now here we will see that GAG is substituted by GUG. So this glutamic acid has been replaced by valine, and this is the normal one. This is the mutant protein. This causes hemoglobin molecules to stick together, forming rigid fibers inside RBC, and they have changed their shapes to sickle cell. So the mutant hemoglobin, what it will do? It will undergo this mutant protein, it will undergo polymerization, that is formation of polymers under low oxygen tension. Thus it will cause a change in shape of RBC. So it is undergoing polymerization, and after undergoing polymerization under low oxygen tension, it will cause a change in the shape of RBC, that is from biconcave to sickle shell. They will be changed. Okay, so this structure change is due to the polymerization of this mutant hemoglobin molecule. Okay, is it clear to all of you now? Now we can talk about a few things which will help in the curing, or we can say that just helping them to give some treatment in case of sickle cell patients. So what are those? So first of all, what is the diagnosis? So it can be diagnosed by blood test, by hemoglobin, electrophoresis and all. So what are the treatments of this sickle cell anemia? Actually medications like hydroxyurea reduces the sickle cell formation. So this medicine, it reduces the formation of sickle cell. Now blood transfusions can also be helpful because they will increase the biconcave normal shaped RBC inside your blood, and which will reduce the deficiency of oxygen in anemic patients, or RBC in anemic patients. Now bone marrow transplant is a potential cure. Now future prospects, we are seeing in the future some gene therapy can be done so that the mutant gene can be replaced with a new gene. So that gene therapy has not yet come. So in the near future we think that this gene therapy is a possible cure for this sickle cell anemia. So this is all for this today's episode. It will be very useful for not only NCERT students but also for pre-medical needs students, and also please do go through all the episodes, all the lessons which I am teaching, and please do subscribe to my channel. Don't forget to share it with your friends. In the next class we will be studying about phenylketonuria and thalassemia. So still then stay tuned to my lesson, and don't forget to write the comments in the comment box. Thank you. Meet you in the next class, and stay tuned to my channel.